Welcome everyone. Sorry for the short delay. We have a high volume of people trying to call in, so we wanted to wait just a little bit. But welcome to the first webinar in the ACS AGRO Division's 2015 to 2016 webinar series, which is sponsored in part by ABC Laboratories. This series has had several moderators: Laura McConnell at Bayer Crop Sciences, Steve Duke at USDA ARS, Julie Ebel at Critical Path Services Knoll, and myself, John Clark, at the University of Massachusetts Amherst. And I'm going to be the moderator today of today's webinar, which is entitled "It Takes a Team: Insecticide Discoveries, Toxicological Problems, Approaches, and Enjoying the Unexpected," which is going to be presented by Dr. Keith Wing. Dr. Wing recently retired from DuPont and is the 2015 recipient of AGRO's ACS International Award for Research in Agrochemicals, which is sponsored by DuPont Crop Protection. Keith received this award for his research and exceptional accomplishments in metabolism, bioavailability, and biochemical modes of action research. Dr. Wynn is currently a consultant in industrial biochemistry and strategic planning, specializing in agriculture and renewable chemical areas. His bachelor's degree was from UCLA, and he received his PhD at UC Riverside with Bruce Hammock, and then went on to do a postdoc with John Cassett at UC Berkeley. Keith was a senior insect physiologist at Roman Haas Ag Chemicals for 9 years and helped discover a novel class of non-steroidal molding hormone agonists, leading to the commercialization of 3 insecticides, including methoxyfenozide. In 1990, Keith moved to DuPont Crop Protection and continued work in agrochemical discovery as a senior research associate, where he helped to discover and optimize indoxacarb, the first commercial insecticide bioactive sodium channel blocker, and later he then discovered Cyazypyr, a novel diamine ryanodine receptor activating insecticide. From 2004 to 2011, Keith worked as a senior research associate in DuPont Central Research and Development, leading a team in the development of a reverse transcriptase polymerase chain reaction-based rapid Listeria detection kit. Following this work, he led a large team for 6 years developing an integrated biomass to biofuels process, which has contributed to the ongoing DuPont cellulosic ethanol business venture. From these accomplishments, his talk today will reflect on 3 insecticide classes which his teams were able to discover, chemically optimize, and eventually register for commercial products. It is hoped that this presentation will stimulate reflections on how to go about finding novel, valuable new crop protections in the futures. With that introduction, I'm happy to introduce Dr. Keith Wing.
Oh, thank you for your kind introduction, John, and thank you to all the attendees for phoning in. So as you can see, the talk title is "It Takes a Team: Insecticide Discoveries, Toxicological Problem Approaches, and Enjoying the Unexpected." First, the most important slide of this whole thing, I think, is actually the thank yous. First, my wife Joan, my kids Allison and Grant, and the extended Wing family. The symposium co-organizers, my friends Jerry Green, Beth Lorsbach, Tom Sparks. Jerry is an independent consultant, formerly of DuPont, and Beth and Tom are at Dow AgroSciences. The ACS AGRO Division and Awards Committee, who have been kind enough to grant me this award. Much gratitude to the award sponsor, DuPont. The previous international award winners, many of whom are close friends of mine, and some of whom actually spoke in my symposium, and the assembled symposium speakers who were kind enough to contribute with me, and most especially my industrial teammates, many of whom will be named further on in the presentation. I just want to review my formative years. I have much gratitude towards former mentors like Franz Engelmann, who got me interested in insect physiology at UCLA. Very, my dear friend and mentor, Bruce Hammock, who I worked with at UC Riverside, and then we moved the lab to Davis around the 81 time frame. And then, of course, the great mentor and friend of many of us, John Cassett, where I spent about 1.5 years in his Berkeley lab, and certainly 1.5 years of the best years of our lives. So And special thanks to those same people. I also want to point out, besides Bruce and his entire lab and staff, Tom Sparks and Vince, who've been really longtime friends, who I still keep in close touch with, and the many friends in entomology and the biochemistry faculty at both campuses. And then at Berkeley, of course, there's a number of friends that I have listed here in John's lab. Some of them, like Louis, Joseph, Derek, Marion, Seaman, Bill, Andy, Mark. I still see them sometimes, but all of them are my friends and alumni of the ECTL, and it's certainly a community that's very close to my heart. I enjoy seeing them when I do see them. So if we get into the main part of the talk, we can reflect on what makes agrochemical discovery a gratifying career endeavor. First 4 bullet points are sort of all things that we've probably experienced at different times. I'm sure we have. Successful agrochemicals are still a very profitable business if we find the right compound. What's interesting to me is that there doesn't seem to be any set way of doing it. It's a mixture of good science, good judgment, experience, and a certain amount of luck. I mean, at times we cannot not acknowledge that latter thing. And some of it can be made systematic or process, such as many of our screens, whether they're biological or things like environmental fate types of screens. The general workflow planning as we go from discovery to optimization to launch can be made into a general process loosely, and then routine chemical analoging programs where you're, let's say, dancing alpha groups around an aromatic ring or something like this, and they're fairly similar chemistry. That can be sometimes somewhat routine, although most chemists would sometimes say that it doesn't always end up that way. But there are a lot of really important aspects about discovery that are difficult to systematize. One of the things that I've reflected on is the inspiration and discovery of the initial lead compound. How does this happen? I mean, most of the chemists, people with chemical backgrounds that I'm aware of, look at these different inspirational sources or a mix as the leads for the compound. And oftentimes it's a very nonlinear process to get to that magic lead that gets the whole chemical analoging series started. And what's interesting to me, too, is that unusual problems often arise during discovery or optimization. That makes it very challenging, but also very interesting and fun. And there's also a few general physical property rules that one could invoke. But if you look at what agrochemicals are asked to do, the requirements are incredibly varied. So if, for example, you look at Chris Lipinski's general rules that we know of for making an orally bioavailable pharmaceutical on the far right column, you can see from molecular weight, log P, number of hydrogen bond donors and acceptors. I mean, of course, there are going to be exceptions to Chris's rules, but they're not at all bad guidelines for making an orally available drug. On the other hand, if you look at what Colin Tice has described in the second column over, a friend of mine from Roman Haas, you can see that whether you're looking at a herbicide as in the H or an insecticide in the I, the physical property requirements can really, really vary. And that's not surprising considering we're asking the thing sometimes to, to last and be very, very long-lived on foliage, or sometimes we're asking it to be mobile after soil application and all these other different things. So, let alone fungicides, which also span a broad range of physical properties. So we're asking for these different compounds to do very, very many different things. So it's hard to kind of generalize with regards to just physical properties alone. So what I'm going to do is in reverse chronological order. All right, so these 3 case histories, each one needed insights into agrochemical ADME, which, as you know, are absorption, distribution, metabolism, and excretion, to be successful. Each built on fundamental science as providing absolutely critical insights into what was going on. And later on, each contributed back to fundamental science, including at the respective target sites. So even though there's going to be some talk about ADME in this talk, there's going to be a lot of relationship back to the target site to toxicologists like myself and many of our friends are all intertwined. So the first section is going to be about the discovery of cyanotrilinoprol, or Cyazypyr, which, by the way, I did not discover cyanotrilinoprol. It was discovered by other chemists in DuPont, and the work was done by a large team of people, and so that'll be described very briefly here. But anyway, it's an expanded spectrum ryanodine receptor insecticide that many of you are probably familiar with. In this history, chlorantraniliprole, or Rynaxypyr, was discovered first, and this is a list of some of the team members that contributed richly to this whole enterprise. George Lahm and Tom Stevenson, Tom Selby, John Freudenberger worked a lot on the chemistry, Dan Cordova and Eric Benner and many others worked a lot on the mode of action. So a lot of people contributed, and this was really, really amazing set of discoveries that has been described, both written and spoken, at many, many different venues. So I'm not going to go into this in great detail. But as you know, chlorantraniliprole is on the market and doing very, very well. And then it was another compound, flubendiamide, which has been on the market from Nihon Noyaku and Bayer. It's also a very successful compound in this area. One, chlorantraniliprole, is a anthranilic diamide, and the flubendiamide is a phthalic diamide. So that provides the background. But after we had discovered chlorantraniliprole, we wanted another analog with improved plant movement, broadened insect spectrum, and so we were wondering how to go about this. We knew we'd have to lower the log P, but we also needed to maintain strong binding or activation of the ryanodine receptor to maintain good insecticidal activity. We need to improve plant movement in the xylem and hopefully the phloem. And importantly, the compound had to be orally bioavailable to sucking and rasping insects. So this is just a listing of some of the cyanotrilinoprol discovery team. In essence, many of the people who work on chlorantraniliprole also work on cyanotrilinoprol. So this is just some of the folks, but quite a few of them had a prominent role in this whole program. So the discovery team was assembled to pursue this problem. So chemists synthesized new structural classes, biologists screened analogs against chewing, but especially sucking insects. So they did a lot of work trying to monitor contact, upward systemicity, and other insecticidal activity against things like whiteflies and aphids and sucking bugs, things like that. Our EFADE team members measured analog physical properties and environmental degradation. What I was trying to understand was plant movement as it related to insect efficacy and how we could get a better analog. So some of the tools I used were LC triplet MS, bioassays with sucking insects, and then measuring xylem upward movement from the roots, phloem backward movements from the leaf tip and downwards, and then translaminar movement, and sometimes I do autoradiography on C14 label advanced candidates. So we cycled through many analogs, we all talked, a lot of team communication. And after much effort and ideas, cyanotrilinoprol eventually did emerge. Now this series of figures is from Barry et al. 2015, Pest Management Science, but it essentially reflects the same kind of work that I did earlier on. Barry's article deals with formulated C14 labeled cyanotrilinoprol, whereas I was only working with C14 labeled technical material, but the principles are the same. What this shows is that if you put cyanotrilinoprol on the petiole of a leaf, it can move outwards through xylem very, very well. So that mobility is well documented in this slide. So from day 1 to day 3 to day 7, you can see the increase in the phosphorimeter, increased compound moving out towards the leaf tips after application of the petiole. So I also did a lot of work on phloem. I also did a lot of work on phloem movement, but they were not. These compounds were nowhere near as phloem mobile as some of the standards that I used, like thiamethoxam, and that frustrated me no end. I mean, to keep the receptor happy, we had to add certain structural requirements, but in order to make it more flow mobile, for example, we would have had to change the molecule a lot, and it was hard to keep both things happy, so we ended up with cyanotrilinoprol, but that ended up being a pretty reasonable compromise. All right, so after a lot of focused work, we were able to go from fluorotriniprol to cyanotrilinoprol, and that compound had much improved plant mobility versus Rynaxypyr, and improved homopterin, emitrin, coleop, and thrip spectrum, and it was very, very much improved, that spectrum. It's still a pretty strong ryanodine receptor activator, still very active on LEPs, not quite as active as fluorotriniprol at both the ryanodine receptor level and in terms of potency of whole Lepidoptera, but still very safe to mammals in the environment, just as like fluorotriniprol was. And actually, Tom Selby covered cyanotrilinoprol in detail during the symposium. And besides the background review articles that he and others have written, we'll defer that discussion to later. But what I did want to describe was that we did learn some things from the cyanotrilinoprol program. A small improvement in plant movement and oral bioavailability to sucking insects can make a big difference in potency. Some of the insect species that we were able to pick up in a practical agronomic sense were now available to us, whereas with fluorotriniprol they were really, really difficult to access, these sucking insects. A balance of driving insecticide physical properties for plant movement with good binding to the ryanodine receptor was necessary. It wasn't easy, but we ended up with what we thought was a reasonable compromise. LCMS and autoradiography were really useful for the plant movement program. And just like everything, good teamwork led to an expeditious optimization program. Now I'm going to switch gears and talk about the discovery of indoxacarb, which also occurred in DuPont crop protection. This is a bioactivated sodium channel blocker insecticide. And this slide shows some of the indoxacarb discovery team. Not everybody, but these are a lot of people that played a key part and really, really worked hard. Most of the chemists up in the top, and mostly biologists and mode of action people and toxicologists in the second group. But hey, there are a lot of people that I haven't named that worked on this project. It was a huge team effort. This particular problem was not one of pure insecticidal potency, but more of selective toxicity. There had been patent inspiration from Phillips Dufar and Roman Haas, TH6041 from Phillips Dufar and RH3421, which came out of the work of my friend and colleague Richard Jacobson and Roman Haas. But these compounds, although they were absolutely superb insecticides, especially on Lepidoptera and some coleops like Colorado potato beetle, they have cumulative and mammalian toxicity. So, for example, you can get 100% mortality rats at 50 mg per kg per day. It's oftentimes a cumulative toxicity. They were quite persistent in the environment. And, I think most of us viewed them as probably not registrable for ag. So how do you get from there to something with good insecticidal activity but dramatically reduced mammalian tox, little accumulation, environmental degraded, acceptable cost and profitability profile, and something that obviously is registerable? So it's a big challenge. A lot of people in the agrochemical industry were trying to work on it. In DuPont, a discovery team was assembled to look for analogs with this improved mammalian and environmental safety, but still the excellent pesticidal activity. So chemists synthesized new structural classes and analogs. Biologists screened the analogs against a lot of insect species. Our ethate members measured physical properties and environmental degradation. I wanted to help improve selective toxicity, so I developed small-scale in vitro metabolism assays in mammalian and, when needed, in insect preps, and then we would do in vivo assays later. I did some binding assays and occasional structural suggestions, and as a team, we all developed a robust SAR program, and there's a lot of close teamwork with everybody. So just like before, we cycled through a lot of analogs, talked a lot on a weekly basis, and you can look at the chemical evolution of this whole class, and it's really quite a lot of work over many, many years by many dozens of people. So again, as I mentioned before, it started with these dihydropyrazoles, which were really remarkable potent compounds. that led to some upside-down dihydropyrazoles. Those eventually led to indazoles and oxindazoles. Those eventually led to semicarbazones and gratazines, and then eventually, especially with contributions from Gary Annis and Steve McCann, we got to trizines and oxidizines, and it was all quite an evolution taking place over quite a long time. It took a lot of creativity by the chemists. Where I helped out was with some toxicologically based thinking about the urea and substitution and what it might do to help us with selective toxicity. So after a lot of ideas, we found an oxidizine that seemed to meet our varied criteria. So that's JW062. So you can see it's an oxidizine with a carbomethoxy group on the nitrogen. If you take radiolabeled JW062 and feed it to Manduca sexta larvae, that's a tobacco hornworm, and then later on extract the radiolabel, you'll see it's almost all converted to a metabolite. In the bottom panel on the right is a peak where I spiked in JW062, and you can see that this, no, I'm sorry, on the bottom, the left peak is a JW062, and that I spiked in, and I was just trying to show that the two peaks are distinct. Now, what is this metabolite? Turns out that it's a decarbomethoxylated metabolite where hydrogen replaces the carbomethoxy group. We were able to see that same metabolite by mass spec in several different Lepidoptera, and to everything I could see, Lepidoptera seemed to convert the compound over very, very rapidly. Other orders of insects do it also. It seems to be slower in other insects, but there does seem to be a pretty good correlation between the appearance of the metabolite and toxicological symptoms in all insects. So as it turned out, the story was dependent on both selective metabolism and chirality. So you can either take indoxacarb, which is a 75% S, 25% R enantiomeric mixture, or JW062, which is racemic 50-50. And both of them have low activity on sodium channels. But once insects bioactivate it, and that's done apparently through esterase or amidase enzymes, it occurs rapidly in Lepidoptera, midgut and fat body, and many subcellular fractions. You decarbomethoxylate this stuff. I didn't see any chiral selectivity in terms of the activation process. It becomes active sodium channel blockers, and only the S enantiomer at that chiral carbon shown here is the active sodium channel blocker. The mammals seem to be able to degrade it. This is a story that was richly fleshed out by our registration and development group, as well as the toxicology group at DuPont, to many small molecular weight metabolites, often through P450 metabolism. So it ended up being a very, very interesting toxicological story. So in terms of mode of action, my good friend Vince Salgado in 1990-1992 did the initial sodium channel mode of action work on pyrazolines. That set the basis for the entire field. And later on in 2003, he also followed that up with work on indoxacarb and DCJW. And they show the same mode of action, which is a sodium voltage-dependent sodium channel block. So if you look at spontaneous nerve activity in the upper section in American cockroach, either in the crural nerve, the central nervous system, or the circle nerve, RH3421 will very potently tamp down or block spontaneous nerve activity. The same thing can be shown with the DCJW or in vivo administered indoxacarb and the Lepidoptera frugiperta in their CNS. So pretty clear indication that we're inhibiting sodium channel-dependent spontaneous activity in the nerves. Dave Sutherland's lab played a prominent role in figuring out the mode of action of these dihydropyrazoles early on. Darlene Dieter and Greg Payne in his lab showed that whether you're looking at sodium-22 uptake in the left panel or tritiated batrachotoxin and binding on the right-hand panel, that the dihydropyrazoles are acting on sodium channels, but at a site that is clearly different than alkaloid activators like batrachotoxin. And they showed that dihydrocortisol binding is non-competitive with alpha-light activators, and also, very importantly, that dihydrocortisols have a very slow off rate, and we were able to observe this in other experiments subsequently as well. So Mike Schnee, my colleague at DuPont, also saw a very potent voltage-dependent block of sodium channels in Manduka larval preparations. In this case, he was looking at compound action potentials. This is work that we published in 1998. So here he looked at the fifth abdominal ganglion from Manduka, stimulating up here and recording here. And what you'd see is a compound action potential, which is a result of many sodium channels contributing. If you administer DCJW, the activated metabolite that I described before, you'll see a time-dependent block, and it's not too slow. Within 15 minutes, it's complete, abolishment of the compound action potential. And if you monitor what's going on toxicologically and look at different enantiomers, both in whole organisms, this is whole lep larvae shown in this whole right-hand column, versus in vitro, what's happening to caps in the left-hand column, you can see that the S enantiomer in all cases is very, very active and very potent against both the whole animal and the caps. But the R enantiomer is pretty much inactive in vivo, but about half as active as the S enantiomer. Sorry, it's completely inactive in vitro, but about half as active or so in vivo as the S active enantiomer. So the whole story fit together quite nicely, both activation and then subsequent blockage of the sodium channels. So the story that's been built up over time that Vince and many others have contributed to is that DCJW and other sodium channel blocker insecticides block the sodium channel in a voltage-dependent manner in local anesthetics like 10. So this is adhering to the nomenclature that Bill Catterall, University of Washington, has helped establish. And we believe that sodium channel blocker insecticides act in a manner that is very, very analogous to local anesthetics like lidocaine, things like that. This shows the different transmembrane binding domains where the sodium channel blocker insecticide might block some of the amino acids that it might be binding to. And more recently, a 2.7 angstrom crystal structure has been solved for bacterium, voltage-gated sodium channel by Bill's lab. And so here's the top view of the sodium channel as pictured. And it, in fact, also describes what may be a phospholipid path shown here in the top and here in the side that might indeed be the pathway by which sodium channel blocker insecticides can access the core of the channel and thereby eventually lead to block. So DCJW and other sodium channel blocker insecticides do block sodium channels in a voltage-dependent manner. There's a selectivity filter that TTX, tetrodotoxin, blocks. It's thought that sodium channel blocker insecticides block more from the interior of the channel, and the toxin and pyrethroids bind at other places and exert their effects. And then by their voltage-gated action, the SCBIs can block the channel, but it doesn't happen with all sodium channels. The more higher or more positive the resting potential, such as is found natively in sensory nerves, can lead to greater block susceptibility, or you can artificially depolarize the axon, and then it becomes more susceptible. But if you got an axon that's sitting at, let's say, minus 70 or minus 80 millivolts or something like this, it may not be depolarized enough to be very susceptible. It's only when you depolarize it more that it becomes more susceptible. And what happens is that apparently it stabilizes, the blocker stabilizes the slow inactivated channels with very, very low off rates. Our compounds really come off slowly and gradually draws more channels into the state. And you can look at indoxacarb also in the field, and it controls a large number of Lepidoptera. It's been a reasonably successful product, and we're quite proud of the work that is done not only on a fundamental basis, but what it's done out in the field. It's also quite active on certain Coleoptera, And then in the hemiptera, it's also been pretty active on Lygus bugs and certain other ones, and Poasca as well. So overall, a very good, solid, and now safe broad spectrum insecticide. So what's been learned from the SCBI and Toxicological program? Well, we think that favorable and selective toxicity can be built into a potent insecticide class. Physical property changes can dramatically change bioavailability to the enzymes and receptors in insects versus mammals. The right team mindset is very important. That priorities will shift from lead area to lead area and should drive the project. And is your organization flexible enough to follow up what those key problems are and do something about it? And very importantly for us, it turned out that the SCBI target site was pretty tolerant to structural changes around the base amide substructure. It turns out as long as you had a fairly flat planar lipophilic molecule with those that central sort of urea thing in the middle, you had some decent shot of structural manipulation around that. That was turned out to be crucial for us. And very importantly to me, the fundamental science helped us solve the problem and establish the SCBI class as a useful class of insecticides. So I want to credit the dozens of researchers that have eliminated metabolic enzymes. Terry long ago, Renee Fireeisen and Jeff Scott, and a number of others that have all worked on metabolic enzymes since then. And then I also want to credit Dr. Narahashi's lab, Dave Sutherland's lab, and Bill Catterall's lab for helping us understand more about the sodium channel fundamentals. So eventually a favorable balance was struck between all these different elements, and we were able to go from a cumulative mammalian toxic class of chemistry to a compound that is still an excellent insecticide, but was dramatically reduced mammalian tox, little accumulation. No, we did not totally abolish mammalian toxicity, as you can see, but they're very much safer than the original dihypropyrazoles, and formulations are even safer. My friend Dan Lin and others worked a lot on degradation of the soil, and we were able to achieve that also, which is very important. Now it's an important registered product. And in addition, there's another compound in the area, metaflumizone, which is commercialized by Nihai Noyako and BASF and Fort Dodge Animal Health. That is another sodium channel blocker insecticide in this whole family, and is now commercial as well. So I'm going to switch gears once again and talk about the discovery of the Bissell hydrazine and Hydrazine agonist. This work occurred at Roman Haas Agrochemicals, and this is a very important part of my life and some of this discovery team that I show here. I still keep very, very close contact with Glenn Carlson and Dennis Patterson, and I see the other folks every once in a while. But we all worked hard on this stuff, and it turned out to be a very interesting story. So a bit of history as we go into this. In 1983, I developed an interest in the fundamental science of ecdysone as an insecticidal target area. Glenn and I had set up a collaboration with Carol Williams' lab at Harvard Biological Labs. And through Carol, I met Lou Safranek, who is an incredible insect physiologist who knew a lot about Lepidoptera and physiology of molting and development. Peter and Lucy Cherbis, who were fantastic cell and molecular biologists, who introduced me to the Drosophila KC cell system. Lynn Ridderford and Jim Truman, by that time, of course, were all already at the University of Washington, Seattle. But fantastic developmental and molecular biologists, and Jim in particular helped to illuminate a lot of things about the ecdysis system and the peptides and hormones that are involved in that system to promote proper ecdysis behavior under the control of the molting hormone ecdysone. And then Dennis Horn of CSIRO and Renee Lafont helped illuminate a lot of things about ecdysone chemistry and metabolism. So a bit of history. In 85 or 86, my chemistry colleague Adam Su had targeted some monobenzoylated hydrazines, and during his reaction he got a mixture of both mono and dibenzoylated impurities, separated them, and had each screen. That whole story is described in his article in 1991. About that same time, shortly thereafter, when we got the results from the screens, it showed one of the compounds, just the dibenzoylated impurity, showed delayed toxicity to lepsin in the screen. One of my biologist colleagues suggested that it was acting as a diacyl urea molting inhibitor like Dimilin, I looked at the structure and symptoms, and it looked a little different. The problem I had was, what's the mode of action of these compounds? Can anything commercial be developed from it? Obviously, that's a consideration for all of us. So, looking more carefully, I noticed that the defective molt was induced very rapidly compared to diacylureas, and decided to test RH5849 as a possible Dicyn agonist or inducer of endogenous Dicyn. And you can see the structural differences between the two classes, especially the bulky T-butyl group and the reversal of the nitrogen and the carbonyl. And the results were very, very surprising. Now, what I want to do is go through a little bit the mode of action of steroid hormones, which, by the way, was illuminated quite a bit by insect work in Diptera very, very long ago in the 60s and 70s. But basically, a steroid hormone like Dicyn, but it could be true of progesterone or estrogens or anything that works by this mechanism, will bind to a steroid hormone receptor. In this case, it's a combination of the Dicyn receptor, ECR, and its cognate necessary partner, ultraspherical protein, or USP. The two bind, and then after that, they can enter the nucleus. The complex binds to specific receptor sites on the chromatin and activates very specific mRNA transcription. The message is transported to the ribosomes, specific proteins are translated, but what you see at the end is physiological responses, which are really profound. I've simplified this a great deal, but there's thousands of reactions occurring, obviously. In Drosophila KC cells, 3 signs of this is to halt proliferation, to induce acetylcholinesterase, and to form long processes. In whole insects, it's a molt, which, well, that's one physiological response to Dicyn, but that molt is composed of thousands of different biochemical reactions. So I've simplified this. So if you look at what happens in Drosophila cells, KC cells, RH5849 is indeed an agonist, but the native steroid molten hormone, 20-hydroxyapatite, is about 100-fold more potent. So whether you're looking at cell process elaboration greater than 70 microns, or cell density, decrease in cell density, or acetylcholinesterase induction, the native hormone is about 100-fold more active than the little agonist that we found. If you look at the receptor level, 20-hydroxyapatite is about 30x as potent as RH5849. I did scattered analysis, and it showed that it's clearly competitive. The tritiated finasterase is a tritiated disteroid, which is a little bit more active than 20-hydroxyapatite. The on rate, Ka, is slowed, but the off rate, small kd, is unaffected by RH5849, indicated competitive binding. And very interestingly, cells made resistant to one or the other don't elaborate processes when they're treated either with what made them resistant or the opposite partner. So there was cross-resistance to both, and both showed reduced levels of binding to tritiated finasterase A. So it appears that 5849 is a weak Dicyn agonist, but what's it doing to whole Lepidoptera or their tissues? This is a photograph of 3-day-old fifth instar of tobacco hornworms. The guy on the top was a control. He got fat and healthy, gained a lot of weight. The poor guy at the bottom had been treated with RH5849 in the diet, and you can see that there's an extra head capsule that's been formed behind 3 days later, and that poor guy was forced to stop eating it 24 hours after treatment, so he's out of luck. So if you look physiologically, the top 2 rows are Manduca sexta abdomens. In L41, they have a high level of juvenile hormone. In L583, they have a very low level of juvenile hormone. Either way, the source of the 20-hydroxyapatite is absent. And if you look on these top 2 columns, obviously RH5849 is much more potent at inducing dicynergic signs. There are arcane little things that we don't have to go into in detail. Just take my word for it that RH5849 is much more active than the native hormone is at inducing these dicynergic signs. And if you either inject or feed orally those same L5 day 0 larvae, you can see that the difference in potency is even more marked, especially by the oral route. And this is consistent with the story. In solid circles, we see RH5849. They stop feeding after less than a day, whereas the other guys keep on gaining weight and eventually molt. And I was able to get McDyson antibody from Skip Bolenbacher at the University of North Carolina and put together a radioimmunoassay according to literature procedures. And if you can monitor steroid hormone levels in the animal while it's been treated by the RH5849, and you can see controls go ahead and show a peak and then a decline of native hormone. And at the peak, they slip their head capsule and get ready to molt. Well, guess what? The RH5849 guys did that a lot earlier at about 24 hours. And in fact, their subsequent levels of steroid hormone are suppressed. So what we think, or I'm pretty sure, is that successful eclosion hormone release and peptide hormones to initiate Ecdysone synthesis are inhibited. So the ADME part of it is that if you feed 10 micrograms per gram of 14C-label RH5849, you can see high enough levels definitely to initiate apolysis, but they slowly decline, and 3 micromolar of the agonist remains in the hemolymph. These are the levels of 20-hydroxyecdysone at their normal peak, and even though normally at the trough 20-hydroxyecdysone is pretty much zero, there's almost nothing that can be detected. So, so the developmentally premature animal initiates apolysis, but it's unable to successfully complete ecdysis, and later on it was found out that eclosion hormone and Ecdysone triggering hormone are not successfully released when they're treated with this agonist. And this head capsule covers up the mouth parts and prevents feeding, and then you'll form cuticle traps the animal. So oral bioavailability and metabolic stability are both critical to toxicity. And how is this happening? So the Bissell hydrazines not only cause premature apolysis of the epidermis and cuticle, but also profoundly disrupt the sensitive axis of neurohormones that the animal needs for correct molting. So prothoracic tropic hormone synthesis in the brain to stimulate Ecdysone synthesis in the prothoracic glands is blocked, and the hormones to release ecdysis behavior are also blocked. So this is a time course of what's happening. This is work by Mike Adams and Zitnon from his lab, and the peak of Ecdysone occurs about here. When this high level of the agonist remains in the bloodstream, a lot of the downstream processes that would normally occur upon the decline of ecdysteroid are not allowed to occur properly, so proper peptide hormone release and synthesis and release are not allowed to occur. There's a feedback inhibition on Ecdysone receptors in neurosecretory cells in the brain and nervous system, and again, these neurosecretory cells are not released properly. So the potency of the 5849 analogs roughly correlated with their ability to displace 38-fast RNA from KC cells. Later on, I and other people got lepidopteran cells like from Flodia, Punctella, and other species, and the correlation was even stronger. But you can see there's a pretty good rough correlation. It indicates that the site of action on Ecdysone receptors is likely the toxicologically significant mode of action in LEPs. It indicates that for the majority of 5849 analogs, they're probably fairly stable metabolically, although I've shown that they're not completely stable, and they have fairly similar ADME and LEPs, and so therefore the fluence of potency is largely influenced by receptor potency. So several thousand analogs are made and characterized at Roman Haas, and the ECR assays were useful along with full insect assays in providing Roman Haas structure activity. So Roman Haas, which is now part of Dow AGRO, was able to commercialize 3 Bissell hydrazines, and then subsequently Nippon Soda and Jiangsu Pest Research Institute were able to commercialize 2 others later. So the ones that now passed through Dow AgroSciences' hands were tebufenazide, elafenazide, and methoxyfenazide, but these were all discovery of Roman Haas. This chromosfenazide was found in Nippon Soda, and then again this fufenazide was found in Jiangsu, all commercial compounds. ECR USP is a nuclear steroid hormone receptor. It was cloned and sequenced first by Mike Cole in 1991 in Dave Hogus' lab, but since then several insect ECRs and USPs have been cloned and studied. They're very, very modular receptors. It's very, very interesting how these things are put together. So the C region, the binding domain that binds DNA, is very highly conserved for all steroid hormone receptors. This ligand binding domain, E, is very specific to the ecdysones and Bissell hydrazines and ecdyson receptors, but one could, it's very, very common for microbiologists in this field to make chimeras in this whole area. So you could, if you wanted to, take out this ecdyson receptor region and put it in the region for estrogen receptor or vitamin D receptor, glucocorticoid receptor, whatever you want, and you'd still have a pretty good functioning receptor that would now be responsive to that ligand. So very, very nice and modular and manipulable systems. Crystal structure of the ECR and USB has been determined, first by Bissell in 2003, and ligands have been co-crystallized here. So here we see a crystal structure of ECR and USP for Heliothus. These are illustrations that I took from Ron Hill's annual review of entomology article. And this is the major pocket here that contains the ecdysteroid. If you look, it took a lot of people a lot of time to try and figure out how different classes, both the RH5849 or its analogs, the Bissell hydrazines, might be binding an ecdyson receptor versus the steroid hormone. And you can see that their binding domains are not completely overlapping. In fact, it appears that BAH's actual binding domain mostly quite a bit overlaps with the steroid side chain of the ecdysones. Very, very interesting finding. It took a lot of work by a lot of people to find that out. These are a large number of ECRs and USPs, or retinoic acid receptors, that have been cloned. It's sequenced in different types of insects, and there are many pest insects included in this list. So covering this whole target site has stimulated exploration of these new classes. So there's amino ketones, oxazolines by Riogene and Intrexon. Bayer has done a lot of work on these N-amino imidazoles, tetrahydroquinolines by FMC. By the way, these are found to be quite active on some dictro, so that's of interest. And then CSIRO has found some gamma-methylene, gamma-lactams, all of which has stimulated a lot of interest in the field. And undoubtedly there's other classes that have not been disclosed as well. Some of them have been discovered by some of these novel chimeric assays that I was talking about in high-throughput screening format. So, interestingly, in conclusion, this funny little compound here can mimic this, and a favorable ADME and binding at the ECR-USP target induces premature molt. There's a devastating physiological disruption when the insect cannot clear the BAH rapidly, and there have been thousands of corroborative studies worldwide, many of whom have been reviewed by my colleagues like Guy Smagi, Toji Dadiala, Lawrence Steinen, Rob Borman. Lots and lots of people have written reviews. The field apparently has been pretty rich. So what's been learned from the Bissell hydrazine program? Great ADME can surmount mediocre receptor binding. Full insect symptomatology is still valuable, I think, for prepared mind. In terms of mode of action, I say don't worry about what the chemistry should be doing. Focus on what it is doing. I got no end of skepticism and ribbing for proposing that these things were acting to mimic a steroid molting hormone, but unfortunately, eventually it ended up being true. It's been corroborated richly. You have to establish the toxicological link from a receptor to cell to tissue to organism to have a very good story. And again, I credit all the fundamental science that laid the groundwork for helping us figure out what was going on. I do question, could this mode of action or target site still be of value given the right market spectrum? A lot of companies have obviously put a lot of time studying it. Well, perhaps the mode of action is, of course, slower than a very, very potent and safe neurotoxin, but could it be useful in the right arenas? Perhaps so. Slight changes in structure can lead to significant distinctions in order selectivity, and a lot of effort has been put into identifying the precise docking mode of BAHs versus ecdysteroids. And for all of us who work in the agrochemical area, I appreciate the opportunity to buy organic produce if I want to, but this cartoon reminds us of sort of where things stand, and hopefully you guys get a little laugh out of that. But fortunately, the BAH story outside of ag is not yet done. We had speculated in 86 and 90 that they could be turned into gene switches in non-insect systems. And in 1990, Roman Haas hired a number of people, Toji, Reddy, and Rob, and others to advance cloning in the ECRs in different insect orders and develop new ligand chemistries. Reagene was later split off from Roman Haas to pursue the system as a gene switch in mammals. The IP was eventually transferred to the University of Pitt Med School, and eventually it was acquired by Intrexon, which is a prominent synthetic biology company. And development of the gene switch in mammals is progressing well. So one of the prominent gene switches that are being pursued by Intrexon is this compound, Valetimex. You can obviously see the structural similarity to the Bissell Hydrazine insecticides. Very much the same, except that the alkyl group here is now bulkier, and there's an ethyl group instead of a methyl here on this ring. So Intrexon has collaborated with Zeopharm Oncology, working with MD Anderson Cancer Institute at the University of Texas. Intrexon is developing this RheoSwitch therapeutic system, along with a number of other molecular biology products, looking for an autologous controllable system. And one of their first efforts has been to engineer an adenoviral vector that expresses an interleukin-12 that uses this RheoSwitch therapeutic gene switch system. So if a person, for example, has a tumor, you can inject it with this IL-12 construct, and later on, the person can take Valetimex orally. It will stimulate the expression in the tumor of this IL-12, and then it will activate tumor-infiltrating lymphocytes to not only attack that tumor, but other tumors that are systemically around the body. So distant tumors as well might be susceptible to this strategy. And indeed, for melanoma, for breast cancer, it's, I believe, in phase 2, and for malignant melanoma, it's in phase 1. So things are looking quite promising. And if you look at the control that can be exerted by this switch, this is the part that I find very encouraging. Under the control of Valetimex administered orally, you can see that log orders of, depending on whether you administer 100 or 160 milligrams of Valetimex, you can get a lot of, expression of the IL-12. You turn it off, and it's off, and 7 days post-treatment, the compound is cleared, and it goes back to the resting state. It's off. And again, showing this on-off behavior, very, very good control. So I'm very, very hopeful about this whole area for potential gene therapy in humans. So the prospects for ligand receptor gene switches in mammals is very promising. It's adapted from these Bissell Hydrazine BCR-US pairs, and it's an additional societal value from insect biochem physiology and an agrochemical source, which, at the time we had discussed it in Roman Haas, and later on the company ended up doing something about it, so we were very happy about that. So in review, these are 3 case histories that ended up being 3 different IRAC classes, class 28, 22, and 18, plus potential impact on human gene therapy. Bioavailability and anatomy properties are important to discovery and optimization, and that combined with target site and full organism knowledge allowed us to optimize and develop chemistry better. And during the symposium, we heard talks on new anatomy and visualization technologies to benefit agrochemical discovery. Finally, I want to commemorate this talk in memory of 4 colleagues who are very dear to my heart: Dan Lynn and Gary Hollingshouse, who were colleagues at DuPont. I actually knew Gary at UC Riverside. They were both PhD students. And Jan Ollinger, my dear friend from Roman Haas, who was very prominent in helping my career there, and Carol Williams, my friend and mentor from Harvard Biological Labs, all of whom were influential in my career. So thank you for your attention, and I include my email address here. If you have further questions, you're welcome to ask me my email. Thank you very much.
Well, thank you, Keith, for that extremely thorough and exciting path through your career. I found it extremely interesting to follow. This concludes our webinar for today, but Dr. Wing will take any questions that you have after a brief pause for this recording purposes, and I'll read the questions to him. So Keith, we have a couple questions. The first question was, and these were questions that came in earlier in your lecture. The first question is, how could you assess it was still safe for mammals during development and testing periods?
How could I assess what was safe?
I believe it was the first compound that you presented.
Cyantroniloprol, well, I mean, what do you mean? How do we assess that? I mean, we have mammalian toxicity assays that.
Yeah, that's the second question. There's a follow-up question that it says, how could you assess it was still safe for mammals during the development testing periods? And the second question was, have you been running in vitro tox studies in parallel on mammals for all similar products tested?
I guess what the questioners are getting at is, yeah, we would, during the development period, when we had promising candidates, we would occasionally test them in mammalian toxicity, even sometimes for, chronic toxicity. That, I think, is not an unusual practice for the agrochemical industry. I mean, we had in vitro tools, yes, but we wanted to test them in vivo also to make sure that we were making progress and that there was a correlation between what we were seeing in vitro and what we were doing in vivo. Obviously, they're not 2 or 3 year studies, obviously, but something more condensed to make sure we were on the right path. Okay, go ahead, John. Anything else?
Yeah, those are the two questions that have come in so far, Keith.
Okay.
And I'm looking at the time, and we're about 1 hour into it. I'm not sure if we should wrap it up. And Keith has asked me to tell everyone that if you have additional questions, you can certainly talk to him offline and ask those questions in that kind of format. With that being said, I want to thank everybody for their attention. On behalf of the ACS Agro Division, I would like to thank our speaker again, Keith Wing, as well as our sponsor for this webinar series, ABC Laboratories. Our next webinar will be the potential for insect herbivore pest management with chemical ecology, that will be presented by Professor James Tumbleson of Penn State University, and that webinar will be held on Wednesday, November 11th at 11:30 Eastern Time. And with that, on behalf of the ACS Agro Division, the speakers, our sponsor, the moderators, and myself, I'll thank you for your attention, and this concludes the webinar for today. Thank you.